case of our Surabhi, newly joined, as well as Abdus and our Aru. They are also our student come teacher and another student of BPharm, the participant, beloved student as well as the master degree student. Actually, this is the second workshop and we will conducted for 2 days. And my topic is the importance of rodent species in the neurobehavioral study. Actually, you know, that we are suffering from the different type of disorder, and day by day there are so many incidents are going on, like RV case, like the different type of the harassment case, as well as I think you know, according to the WHO in 2019, the 1 people out of 8 are suffering from the mental disorder. And not only that, the last year 43,000 divorce cases already filed. Okay? So actually, why that is occurred? So when we are thinking regarding the matter, due to the memory deficit, due to the impaired memory, as well as when we are suffering from the anxiety, depression, there are different type of psychological disorder. So this is the time for understanding the what is the relation between the which part of our brain like neuron is correlated with the different type of the behavioral disorder. Not only that, that is also when we are thinking regarding the cognitive impairment, as well as the mental retardation, as well as the melancholy, as well as the anxiety, depression, which part are mainly involved in our brain, as well as in case of when we are suffering from the suppose Parkinson's disease, in case of the Alzheimer's disease, in case of the behavioral defect. So, how our brain are mainly the effect, and not only that, when we are using the different type of drug, how that drug are mainly acting. So mainly evaluation for that different drug, we are mainly choosing the rodent as well as the zebra. Why? Because there are the 95% similarity in anatomical as well as the physiological as well as the proteins of the humans. So the similarity is there. That's why we are mainly choosing the rodents. Okay. So I discussed the introduction and why rodents are mainly chosen. Then the purpose of animal model. Then different experimental animal, husbandry, different model to access the neurobehavioral changes, rodent limitation and conclusion. So before some time I already discussed. So we have to know the which part of our brain are going to be related with that. Okay. So why that the time is very short, within half an hour I have to complete. So I will discuss quickly. So how our brain is very affected, and another point also there, which part of our brain are mainly related with our neurobehavioral activity, and when we are suffering from the Alzheimer's disease as well as the Parkinson's disease, memory disease, which part are mainly responsible for that one, and when we are using or go for the treatment purpose mainly, the which part or how, what is the treatment, what is the molecular level of action, what is the mechanism of action. Then another point also there. So why we are choosing the animal? Mainly the animal are very small in size, and that is also similar to 95% of the protein coding genes in mice, and similar to the humans, and the protein coding having 85% similar, as well as the huge amount of the reproduction of the animal, and that is also really the easily available of the animal. That is also essential for the research purpose. And lifespan of the animal also very short. So next one, so we have to, for the rodent, the main purpose of the rodent. So actually, when we are targeted the different type of drug, how they are act, how the that we by which person we are really analyze, and then the mainly we are using the different type of rodent as well as the miscellaneous under the rodent, mouse, rat, guinea pig, zebra, as well as the hamster. In case of the miscellaneous, frog, pigeon, zebrafish, chicken, and in case of the non-rodent, mainly in research field, the rabbit, dog, and monkey are mainly used. Next, there are the different strains of the mouse, and that is also the C57 blacks by 6, then the DBA by 2, DALT by C, Swiss albino mice, you know. So actually, that type of animal are mainly used for the research field, the cardiovascular disorder, as well as the neurological disorder, genetic disorder, diabetic research. So we are using the different type of strain of the mice as well as the rat. Okay, in case of rat, the Twister Tyro, that is as well as the long events and the Sparagius, this type of strain are also mainly used for the research purpose. Next one is the husbandry of the rodent. So you know that the rodent are mainly placed in the cage, the upper portion is open, and in case of the bedding, mainly the corn cob, that is also by the corn cob, and the temperature should be maintained 20 to 26 degrees centigrade, humidity should be maintained 30 to 7 degrees centigrade, and the lighting condition mainly the day and night 12 hours interval. Okay, and in case of the diet, we have to supply the pellet and the water at libitum, means sufficient amount of water also necessary. So mainly for neurobehavioral study, mainly we have to evaluate the different type of behavior, like the impulsive behavior, means that is also the emotional. Okay, emotional behavior. The drug is what is the drug are mainly acting, as well as in case of Alzheimer's disease, in case of the Parkinson's disease, in case of schizophrenia. So we are mainly of that type of experiment through the rodent, and for the different type of the neurobehavioral study, mainly we are using the different type of method. In case of the central nervous system, suppose the depression study, central nervous system depression and the stimulation, we are using the actophotometer. Okay, and the latest version of actophotometer also available in your pharmacology lab. That is also your the IR, infrared actimeter. Next one is your the tail suspension test, as well as both swim test. These are also mainly used for the CNS depression and stimulant activity. In case of spatial memory and latent memory, the different type of model are there, like your novel object recognition, as well as the YS, PMH. And in case of the muscle coordination, actually muscle relaxation or muscle coordination, maybe the rotor rod are mainly used, and latest version of the rotor rod, the instrument also available, that is also grid tank. And in case of the anxiety model, the light and dark model, and the elevated classmates also available. Okay, we have performed the different type of tests, and that instrument also mainly used for the post-swim test, tail suspension test, I have mentioned previously, and the actophotometer, that is also mainly used for the CNS stimulant, and the depression activity, that is also locomotor activity, locomotion, and IR activity I already mentioned, and incapacitator meter, that is also mainly used for the pain and anti-inflammatory, or evaluation of the pain as well as the anti-inflammatory activity. And grip strength, that is also used for the study of the toxin study, as well as your muscle relax, as well as your toxicity study, as well as your hormonal test. Okay, hormone testing of the hormones. So we are also using, and in case of the Nobel of the recognition, by which we are also evaluate the cognitive behavior as well as the spatial memory of the animal. And in case of the elevated plasmids, we are also using by the anxiety, whether the animal are suffering from the anxiety or not. So next one is the force swing apparatus. So actually, when I am telling regarding that force swing apparatus, that is also the tank, water tank, and that water tank should be filled up to 15 centimeter. That is also the water tank. The up to 15 centimeter water should be filled, and we have to place the animal on there. Okay. So when we are placing the animal and the animal are not escaped, okay, and so ultimately that is also when we are placing the animal on the water, we are try to come out from the water. So we are pressurized to swim. So that is also known as the force swim test. Okay, that is also give the depression activity. So ultimately, maybe what happened now, we have to take the reading the last 4 minutes of the 6 minutes. Okay, means why 2 minutes is we have to avoid, because first 2 minutes they are vigorously moved. That's why we have to leave the first 2 minutes and another last 4 minutes we have to evaluate, or we have to take the data. And not only that, when they are movable, that is also the mobile stage. So we have to take the immobility as well as the mobile stage. That means the immobility means they are not moved. So that is also the depression stage. And when the animal is movable condition, that is also in the less depression. Okay. Then another point also there. That is also one model for the anti-depression activity. Another model also there. That is also the tail suspension method. The tail suspension method, we have to hang the tail of the animal to the. That is also the instrument you see. This one, that tail suspension, the second one. That tail suspension, that is the hook also there. We have to attach the tail of the animal to the hook. Okay, and they are also try to move, and movement of their hind paw as well as the fore paw, as well as they are also try to, you see here. Okay. Okay. So their movement, when they are in a movement of their hind paw as well as the fore paw, or the bending of the tail, or one to crawling, so that is also the movable condition. That is also the mobile phase, and we have to count that one for 6 minutes. Okay. The out of 6 minutes, how much time they take the immobile and the mobile condition. So immobile means depression, mobile stage means anti-depression. Okay, we are really evaluate the anti-depression activity. We have to suppose the drug diazepam. You know that is also anxiolytic as well as the antidepressant, as well as the fluoxetine, acetalopram. We may evaluate, or we may also evaluate the another polyherbal formulation. Okay, by which we may evaluate the antidepressant property by this instrument. Next one is your, I already told you, actophotometer and the IR actimeter. Actophotometer, that is also, you know, that are also mainly the light beam is passed from one portion to another portion, and when the animal move, then that is also mainly got the light or beam. So ultimately we have to take the count. Okay, so if the animal is in the depression, they are not move, okay, or move very slow, and when the animal is, the CNS stimulation is over, they are move fast. So ultimately they are also got the light beam. Okay, so totally we have to mainly take the reading. But what is the difference between actophotometer and the IR actimeter? Then IR actimeter are mainly controlled by the infrared. Okay, the light beam are passed, and another point also there, we may also here we may also take the total count. Okay, the total movement. But in here we may count the x-axis as well. X-axis means that is the x-axis, y-axis as well as the z-axis. The x-axis means that is also the front side movement. Then y-axis, that is also mainly the left side movement, and z-axis, that is also mainly the rearing. Okay, and always the animal are the wall hugging in nature. They are move the wall throughout the wall. Okay, and another point also there, we may also using the video recording, and not only that, we are also placing the whole board mode. Whole board mode by which we may also, by using, we may also determine the anti-depression property also. Okay, by using the your no smoking. Okay, that is also the difference between the actophotometer and IR actimeter. All both are available in the pharmacology lab. Then for the assessment of the pain and inflammation, we are using the incapacitometer. Okay, and by which we may also mainly determine the pain and inflammation. So mainly we have to place the windpower of the animal in the some places are there actually. The left and right portion, left and right windpower we have to place, and if they are giving the data in same, means they are giving their total body weight on the windpower. Okay, so ultimately what happened? Now that is also mainly if they are suffering from some inflammation, or they are suffering from pain, or they are suffering from some aging purpose. So ultimately they are not give the equilibrium weight in both the windpower. Okay, and sensor also there by which we may also take the reading. And in case of the grip stage, that is also the latest version of the rotarod. Okay, rotarod, you know, we may evaluate the muscle relaxation property. So actually, what is the advantage in case of the grip stage? We have to maybe using the steel plate, and on there the 4 power as well as the different method also there. The percentage of the tension break and the compression break we will also measure, and mainly we are using the effect of the sedative as well as the hormone, toxin, muscle relaxation we may also use. But in case of the rotarod, the total, if we are using the suppose diazepam give the muscle relaxation property. So as early as possible, they are also mainly relax their power and they are fall down. But in case of the grip state, mainly that is not dependent upon the total relaxation of the body. So ultimately they are also the gripping strength of the 4 power as well as the hip power. So in case of the tension break, we have to place the 4 power on that. You see the picture. Okay, that is also the plate made by the steel. and we have to pull the tail, and that is also connected with the sensor, and that reading will be given. And in case of the another point, that is also compression brake stem, so we have to push that one. Okay? By we have to place the forepaw. Forepaw, do you know? Forepaw konta? Forepaw konta? Samne du. Okay? Pinpaw pechone. Okay? So next one is your novel object recognition. The novel, by using the novel object recognition, we may also measure the spatial memory as well as the cognitive memory. Spatial means sthan chihnito kar, means suppose your residence is suppose Bagna. Okay? So you already get your bus here and you cross the Bagna. And when you reach to Meshada, then you go, okay, my home at Bagna. Because you forget your native place or you forget your home. So that is also the spatial memory. That is also the actual place. So in here we have to give that training. Okay? So first of all, the novel object recognition, the instruments are available, and maybe you see here that is also the box and the black box that is also available in your pharmacology lab, and maybe first the 10 minutes first day for 10 minutes you have to give the habituation should be necessary. Okay, and next day you have to place the two same color or two same size object should be placed, and 3 to 5 minutes we have to keep the that animal. Okay, and you have to observe for 5 minutes, and you have to